12:30, na? We can start.
Are you there?
Vandana ma'am?
Yes, I'm there.
Yeah. Can you just put your video on, Vandana madam?
Yes, yes. This is good enough?
Yeah. Good enough. You are audible also.
Right.
Yes, Aksha. We can. It's 4:30. We can start.
Okay. Good evening, all of you. Welcome back. So we'll have Dr. Vandana Panda, Associate Professor of Pharmacology Department, to chair the today's session, the last session, but not the least, of our own principal, Dr. Urmila Joshi. Vandana ma'am, over to you.
Thank you so much, Akanksha. It is my pleasure to introduce Dr. Urmila Joshi, our beloved principal professor and head of the Department of Pharmaceutical Chemistry, Kundnani College of Pharmacy. Dr. Joshi received a B.Pharm and M.Pharm from Bombay College of Pharmacy and her PhD from UDCT, now ICT, in medicinal chemistry under the guidance of Dr. V.M. Kulkarni, a legendary teacher and researcher. She immediately joined KMKCP and has been teaching organic chemistry, medicinal chemistry, drug discovery, and spectroscopy passionately to UG and PG students for the past 3 decades. Her research interests lie in the area of cancer therapeutics with a focus on tyrosine kinase inhibitors, QSAR, and different computer-aided drug design techniques. Dr. Joshi is a recognized guide for M.Pharm and PhD in pharmaceutical chemistry and has so far guided more than 30 M.Pharm students and 4 students for PhD. On a personal capacity, she has received robust funding to the tune of 1 crore plus and counting from different governmental agencies, including University of Mumbai, AICT, UGC, ICMR, DBT, and DST SERB. For the institute, she has garnered a few crores by way of DST FIS grants and multiple industry projects. She has 40 publications in national and international journals to her credit and has received 3 Best Paper Awards for publications and numerous awards for oral and poster presentations at various symposia and conferences. She has also authored 15 articles for a popular science series in Marathi. Dr. Joshi is an evaluator for the J.N. Tata Endowment Scholarship and the Narottam Saksaria Foundation Scholarships in Pharmacy and Technology. She was an editorial board member of IJPS for a very long period of time and is a peer reviewer of many national and international reputed journals. She sits on important regulatory committees of the University of Mumbai and DTE Maharashtra. She is also an external referee for M.Pharm and PhD thesis for ICT, Bharati Vidyapeeth, SNDT University, and NMIMS. I now welcome Dr. Joshi and invite her to start with her talk.
Thank you so much, Dr. Vandana, for this kind introduction. Am I audible?
Yes,
ma'am, perfectly.
Yes, ma'am, clearly
I
audible.
will start sharing the presentation. Can everybody see my screen?
Yes, ma'am.
Yes.
Welcome to this last session of the ATAL FDP, and my task has become difficult by the preceding excellent lecture by Dr. Vandana Patravali. I will be speaking on kinase inhibitors, the road ahead, and thought that I will adopt a storytelling approach. So let us start with the story. Way back in 1959.
Madam, sorry to interrupt you, but if you keep your video off, maybe you will be clearly audible to the attendees.
Okay. I will have to. How do I cannot see it. Okay.
Ma'am, that's on top right corner. There is a video.
Yes. Now is the video off?
Yes, ma'am. Your video is off and you're clearly audible. Thank you.
Okay. Just a minute. I wanted to start with this. So, the story starts from 1959 when Peter Nowell, who was working at the University of Pennsylvania, he was working with cancer cells. And one fine day, when he wanted to prepare a slide for observation under microscope, instead of washing the slide with normal saline, he washed it with tap water. There was a change in osmotic pressure and the cells swelled. And when such cells he observed under microscope, for the first time he could see chromosomes very clearly. He was zapped to see the chromosomes. He was not aware of the existence of chromosomes. There was no relationship which was detected till then about the cancer cells and the chromosomes. But he wanted to know more. So he contacted David Hungerford, who was a PhD student, and he was working on certain leukemia cells. Both of them developed the technique and they could see the chromosomes. And looking at the chromosomes, David Hungerford realized that some of the leukemia cells have an abnormally short chromosome number 22. Since they were working in Philadelphia, they called this abnormal chromosome 22 as Philadelphia chromosome. Further work in this direction was done by Janet Rowley. She identified that Philadelphia chromosome is responsible for chronic myelogenous leukemia, and she also went ahead to prove that the Philadelphia chromosome is formed by translocation of part of chromosome 9 and chromosome 22. You can have a look at this. Chromosome 9, a part of chromosome 9 separates, a part of chromosome 22 separates, and a short Philadelphia chromosome is formed. This was later identified that this gene encodes for BCR-ABA fusion protein. And this BCR-ABA fusion protein was later identified as a tyrosine kinase linked protein. So let us understand what is a tyrosine kinase. Tyrosine kinase is a protein that is activated by phosphorylation. Upon activation, it carries out its functions. But it was realized that this protein, BCR-ABL, is auto-activated and therefore is responsible for aberrant cell proliferation, which was associated with leukemia. So, the next step, obvious and logical step, was to develop inhibitor of this kinase. Okay. Now in late 1980s, a scientist called Nicholas Lydon was at Ciba-Geigy. Now it is called as Novartis, and he led a team for identification of compounds with inhibitory activity against protein kinases. In his efforts, along with his team, he realized that 2-phenylamminopyrimidine is a lead compound. It has low potency, poor specificity, but it has a potential to be developed to a better active compound. So he, along with his team, started the efforts. Look at this. This is 2-aminophenylpyrimidine. Now, the additions that were done, this is a classical example of a very detailed SAR. They attached pyridine to it. It improved the cellular activity. Then they attached the benzamide nucleus. Everybody can see my cursor?
Yes, ma'am.
So I can try having a pointer, so it will be better. They attached a benzamide. With benzamide, the tyrosine kinase inhibitory activity improved a lot. But still, this compound was a mixed kinase inhibitor. So, upon introduction of this methyl group, there was a loss of protein kinase C inhibition and therefore the associated side effects. Now, the only problem remained, and that was improving the solubility and bioavailability. At that point, chemical methods were more popular for improving solubility and bioavailability. I'm sure if Vandana madam had worked at that time, she would have devised newer methods. But at that time, chemistry dominated, and for improved solubility and bioavailability, this piperazine was introduced. This led to a compound called imatinib. Novartis, Ciba-Geigy then went further, testing these compounds in rats, mice, rabbits, dogs, etc. And in 1996, they observed that there were several toxic effects and it caused liver damage in dogs. They were about to discard this compound when somebody thought that let us test in monkeys and in vitro human cells. The results were encouraging. And so in 1998, first clinical trial was planned by an oncologist, Brian Drucker. The oncologist every day came in contact with cancer patients and was getting frustrated because of the deaths which were associated with leukemia. But Novartis was reluctant to supply the drug because of the side effects and other things. Upon pressurizing, finally Novartis agreed to supply 25 grams and the dose was 400 milligrams. So it was just sufficient for about 100 patients. But imatinib showed miraculous results in the first trials. And in 2001, FDA approved imatinib under the trade name of Gleevec. Okay, there is a story. Initially, the company had mentioned Gleevec as a trade name, but FDA officials felt that this is so similar to an anti-diabetic drug. And so, the name was changed to Gleevec. So, with this interesting story, let us understand what is a kinase. Protein kinase is an enzyme which catalyzes conversion of a protein to a phosphorylated protein. Okay, and this reaction is catalyzed by ATP. The reverse reaction of dephosphorylation of protein is catalyzed by protein phosphate. Now, why protein phosphorylation is so important? Because a lot of consequences of protein phosphorylation. The phosphorylation alters the conformation, that is the shape and the function of the protein. Secondly, phosphorylation regulates many cellular processes, including cell proliferation, cell growth, apoptosis, signal transduction pathways, and many more. And this particular consequence of protein phosphorylation makes it very important. Now let us understand which residues are phosphorylated. Essentially, OH groups are phosphorylated. All of you may recollect that serine, threonine and tyrosine have OH groups on their side chains. So these OH groups are phosphorylated. Sometimes histidine also undergoes phosphorylation. that is N-phosphorylation, but the N-phosphorylated structure of histidine is not as stable. So essentially, it is these three, tyrosine, threonine and serine residues. The kinases are therefore divided into two categories: tyrosine kinases, where phosphorylation of tyrosine residue occurs, and secondly, serine threonine kinases, where the phosphorylation is of serine and threonine. Okay. The tyrosine kinases are divided into receptor tyrosine kinases and the non-receptor tyrosine kinases. Likewise, the serine threonine kinases also, but serine threonine kinases, majority of them are non-receptor kinases. This slide shows the serine CH2OH side chain, which is phosphorylated. O gets converted to O PO3H2. Similarly, threonine, the OH gets phosphorylated, and in tyrosine, the phenolic OH gets phosphorylated. So far, so good. Let us go ahead now. If kinase is so important, it is necessary for us to understand the structure of kinase. Okay, the kinase domain consists of two important lobes. I think everybody can see this one part, the N lobe, and the other part is the C lobe. Okay, as you can see, the N lobe is smaller, the C lobe is larger, and this is the hinge region which joins the N lobe with the C lobe. Near the hinge region, in a pocket deep seated, there is a ATP binding site. You know that ATP is required for phosphorylation, so ATP has to bind to kinase. Apart from this, there is a DFG motif. Okay, this DFG motif is part of a loop which consists of 3 amino acids. Okay, we will discuss the details about this later. It is a flexible part. Okay, kinases are known to be flexible in the sense they change their shape, and the changing of 3D shape, there are 2-3 components which are important. One of them is this DFG motif, or these 3 amino acids. The second one is the C helix. You can see the C helix. This can also change its position. Okay, let us see. Because of the change in the position of the C helix, can you see here? The C helix is so much near here, whereas in the next diagram, the C helix has moved away. Okay. Similarly, in the DFG, D stands for aspartate, F stands for phenylalanine, and G stands for glycine. The aspartate and phenylalanine, because of their flexibility, can have two possible orientations. One, in which this aspartate is oriented towards the ATP binding site and phenylalanine away from it. The second orientation is there is a flipping. Okay, there is a flipping or change of the positions when phenylalanine points at this ATP binding site and aspartate points away from it. Okay, now this conformation, the first picture shows the conformation, which is the active conformation. Okay, the active conformation is where aspartate is pointing towards the ATP pocket, phenylalanine pointing away from it, and the C helix is quite nearby the ATP binding area. In the next DFG out conformation, the flipping has occurred So aspartate and phenylalanine have changed their positions, and C helix has also moved apart. Because the C helix has moved apart, look here how much more area is available for binding of the inhibitor. Now, some of you must have definitely realized that there are two important flexible components. One is the DFG and other is the C helix. And because there are two important components, flexible components, we get at least 4 different extreme situations. Okay, here look at the first. Here the DFG is in. DFG in meaning phenylalanine pointing away from the ATP binding site and the other amino acid pointing towards it. Whereas alpha helix is also nearby the ATP binding site. Now in the second case, the phenylalanine is still pointing away. Okay, have a look at this and try to understand that phenylalanine is still pointing away, but the alpha C helix has moved away. Okay, and this has opened up some area over here. Now, when phenylalanine and aspartate flip each other's position, what happens? Look at this aspartate. It is now pointing outwards. Phenylalanine is pointing inwards. Okay. But the C helix is still near the ATP binding site. And in the fourth extreme, where the flipping has occurred, phenylalanine is pointing inwards, but the C helix has moved away. Okay, so C helix in, DFG in, C helix in, DFG out, C helix out, DFG in, and C helix out, DFG out. These are the 4 extreme conformations or shapes in which the kinase can remain. Now this is a video. This is receptor tyrosine kinase, but it is applicable to other kinases also. Okay, what happens is, this is a ligand. There is an extracellular binding site. The ligand binds to it. When the ligand binds, there is dimerization which is occurring. Okay, and this part of the receptor is the kinase part. Okay, now what happens upon dimerization? Look at this. There is a conformational change upon dimerization and phosphorylation occurs. This is the kinase active site which phosphorylates the tyrosine residue. Okay. Now what happens upon phosphorylation? Upon phosphorylation, the kinase is able to get attached to other proteins and phosphorylate them successively. Okay, the kinase or this receptor is able to get attached to other proteins because of phosphorylation. You must have heard of this pathway, RAS, RAF, MEK, MAPK pathway of signal transduction. So, successive phosphorylation, also known as kinase cascade, occurs. And this results in transcriptional regulation. Okay. Now, let us try to understand why kinase and kinase inhibitors are so important. What I have shown on the slide is a cell cycle. All of you must have learnt in B.Pharm the cell cycle. So this is the growth phase. After growth phase, the cells go to the synthesis phase where there is DNA synthesis occurring. After that, there is second growth phase. In the second growth phase, the synthesis of say spindle fiber and all occur. And finally, there is mitotic phase occurring. Okay. Now, all these phases are very tightly controlled by multiple kinases. Okay, first type of kinases are growth factor kinases, which decide whether the cells can go to G1 to S. Apart from growth factor, there are multiple cyclin dependent kinases which also play a role. And then from S to G2 and G2 to M, there are various other kinases which keep on checking whether everything is going right or wrong. Okay, but in cancer, some kinases function aberrantly. They allow everything to proceed without checking, and there is consistent cell division which occurs. Okay, so kinase inhibitors are very important in cancer. We have seen the history of imatinib, but post imatinib there were multiple kinase inhibitors which have come into clinical practice. Now let us try to divide them. We have said that the active conformation of the receptor is DFG in. Okay, so type 1 inhibitors are the inhibitors which bind to DFG in conformation. What do you understand by DFG in and active conformation? The C helix is near the ATP binding site. D or aspartate is pointing towards ATP binding site and phenylalanine pointing away from it. Now, there is something called as type 1.5 inhibitors. Okay, in type 1.5 inhibitors, what happens? DFG is still in the in form, but the C helix starts moving out. Okay, since the C helix starts moving out, the conformation now gets converted to an inactive conformation. Okay, so type 1.5 inhibitors bind to inactive conformation. Type 2 inhibitors bind to a completely DFG out conformation. That means DFG is in out form, phenylalanine pointing towards the ATP binding site, and C helix has moved away. Okay, then there are other different inhibitors. There are type 3 inhibitors which bind to allosteric site adjacent to ATP binding site. There are type 4 inhibitors. They also bind to allosteric site, but the allosteric site is different. It is far away from the ATP binding site. Then there are bivalent inhibitors and there are covalent inhibitors. Okay, so let us try to understand this, how the binding occurs. Here I have shown a kinase in active form. Okay, in active form, the aspartate is pointing towards the ATP binding pocket. This is the ATP binding pocket. Apart from ATP binding pocket, there are front pockets. There is a gate area over here. Okay, and if the C helix moves away, then there will be some back pockets. Okay, whereas in the DFG out form, okay, in the DFG out form, some other back pockets are accessible by the inhibitor. The ones which are shown here in pink. Okay, this is accessible and these back pockets are also accessible. Plus, there is a allosteric pocket which is also accessible. Now, let us try to understand binding wise. The type 1 inhibitor is going to bind to ATP binding pocket, the front pocket and the gate area. Okay. Even 1.5 inhibitor can bind to ATP binding pocket, front pocket, gate area. Type 2B inhibitors also bind over here. But in addition, if it occupies the back left, depending upon which back left it occupies, it can be either type 1.5 or 2. depending upon the position of the C helix and DFG. This we have already said, allosteric site in type 5 inhibitors. It is a bivalent ligand, so it occupies two places, ATP binding pocket and the other pocket. Okay. The next question which comes to mind is, when you look at a crystal structure, there is only one crystal structure with you. So how are you going to decide whether this crystal structure has DFG in or out, whether the C helix is in or out, or C helix is towards the ATP binding site or not? This problem is faced by multiple researchers, and a solution to that is measuring the distances. First is between the phenylalanine and the glutamate of C helix. Okay, and second one may be phenylalanine and the gatekeeper plus 3 residue. That is the residue which is third from the gatekeeper residue. Now there is available a CLIF database which will be of use in assigning the correct conformation to kinase. We will know using the CLIF database whether it is a DFG in C helix in or DFG out C helix out or any other conformation. Now I think all of you must have read about this statement by Tolstoy: all happy families are alike, but each unhappy family is unhappy in its own way. Taking these words from Tolstoy, I wish to state that all active kinases are alike, but each inactive kinase is inactive in its own way. Okay, reasons why it is inactive are different. Now first, let us come to active kinases. Okay, active kinases bind to type 1 inhibitors. The active conformations of the kinases bind to type 1 inhibitors. Okay, so let us understand what are type 1 inhibitors. Shown over here is Gefitinib, which is a type 1 inhibitor of EGFR. Okay, I think everybody may be aware that EGFR is the culprit in about 80% of the solid tumors. EGFR firing is doubled, EGFR activation is much more in about 70 to 80% of solid tumors, and therefore Gefitinib, which is a type 1 inhibitor, gains importance. There is another type 1 inhibitor, Aerolotinine. Okay, look at the structure. This is a quinazoline, and quinazoline is important because it binds to the ATP binding site, and ATP has a purine structure. Okay, so this is similar to that, and so it is important, this structural feature. Then the other features are different. Okay, ATP after this has a sugar and then phosphate. So similarly here, the size of this side chain is similar to sugar and phosphate. There is additional here phenyl ring, and that binds to the receptor in a better way. As I mentioned, type 1 inhibitor binds to the active site of the kinase or active site of the kinase in an active form. Okay. Now, active forms of multiple kinases are similar, and so a type 1 inhibitor, although it is designed for binding to EGFR, can bind to multiple other kinases, and this binding results in adverse effects. Okay, so selectivity is a major issue in therapeutic use of type 1 inhibitors. Okay, active site of the kinase in active conformation is similar because function of every active site is same. It has to bind ATP and it has to phosphorylate the protein. So the function is same, so the structure is also same, and therefore multiple inhibitors can bind to multiple kinases. Mutations under pressure. Typically, after some time of disease-free state are very common in case of type 1 inhibitors. And these mutations happen in such a way that ATP binding is not affected, but drug binding is affected. Okay, and so they reduce the activity of the drug. Scientists have addressed these challenge on using multiple techniques. Okay, selectivity. One way of addressing selectivity was very unique. I mentioned that there is one single gatekeeper residue. Okay. Now, size of the gatekeeper residue varies with different kinase. Okay. If the desired kinase has a smaller gatekeeper residue, then off-target effects can be taken care of. Okay, because a smaller gatekeeper residue will allow binding of different types of inhibitors, and a larger gatekeeper residue will allow binding of different types of inhibitors. Okay, so selectivity has been addressed based on gatekeeper residue. Resistance has been addressed by adding another kinase inhibitor or inhibiting same or the different kinase. An example I will give you. VEGFR inhibitors or EGFR inhibitors. Okay, EGFR inhibitors, I said, are used for solid tumors. VEGFR inhibitors are used to inhibit angiogenesis, that is blood capillary formation, which is required for tumor. Now, since there can be resistance to VEGFR, how the resistance develops is when another kinase, c-Met, plays an active role in angiogenesis. So you inhibit VEGFR, still the cancer cells don't respond because there is another kinase through which they can go for angiogenesis. So if you add inhibitor of that kinase also, that is c-Met inhibitor along with VEGFR inhibitor, then the resistance can be taken care of. Okay, this is regarding the type 1 inhibitors. Coming to type 1.5 inhibitors, okay, they bind to the conformation in which DFG is in, that is aspartate pointing towards the ATP binding pocket, but C helix moves away. Since C helix moves away, this area is accessible or available for inhibitor to bind. Okay, so inhibitor binds to this area also. One example of this is Dasatinib, which binds not only in the ATP binding region and the front pocket gate area, but also extends in the back left of the protein kinase. One more drug of this type is Lapatinib. Lapatinib again is an inhibitor of EGFR. It is type 1.5 inhibitor. Just see, have a look at Lapatinib, just see its shape and try to understand that it will bind like this. Okay, it will bind in fact like this, because this is the ATP binding region. It will bind like this. Since C helix is out, the conformation is an inactive conformation and it will bind to an inactive conformation. Okay, there are some inhibitors which also bind to DFG. Okay, and because they bind to DFG, there is a stronger binding with kinase and therefore greater inhibition. Some people must have wondered how a single compound can be either type 1 or type 1.5 inhibitor in same or different kinases. Okay, we have seen Aerolotinib, we have seen some other compound also, we will come to that in the next slide. What happens is the difference in 1 and 1.5 conformation is the position of C helix. Okay, dimensions of the binding pocket may differ in different kinases. The binding pocket becomes larger in 1.5 and smaller in 1. Therefore, same compound may be type 1 inhibitor or type 1.5 inhibitor. Okay, remember the change in the conformation of a receptor going from DFG into DFG out or C helix into C helix out is not all or none. There are multiple accessible conformations which are possible. Okay, it is not like one person gets highest marks. Since all are teachers, let me give this example. It is not that one person gets highest marks and rest all are all failures. There is a gradation. Similarly, conformations also, there is a gradation. Another example is Bosutinib, which is a type 1 inhibitor of SRC kinase and can be either type 1 or type 2 inhibitor of ABL kinase. Okay. Similarly, Sunitinib and other examples are mentioned over here. Now, Bosutinib has a distinctly different reason. Otherwise, it is the dimension of the area available for binding. Okay, but Bosutinib has a distinctly different region. Just have a look at it. Bosutinib is a type 1 inhibitor of SRC, but it can bind to type 1 as well as type 2 of ABL. Now, why? SRC, the energy penalty that it pays to adopt the DFG out or the inactive conformation is much higher. That means SRC will prefer to remain more in active conformation because its inactive conformation is less stable. Okay. Therefore, what happens is Bosutinib can bind to SRC active conformation, but ABL going to inactive conformation very easy. And so, in ABL, Bosutinib binds to both active as well as inactive conformation. Okay, so the inhibitor chooses the energetically accessible conformation. Here, as I mentioned earlier, all the active kinases are so much similar. The inactive kinases are so much different. Look at this. The D, the F and the G, that is aspartate and phenylalanine, can adopt very few conformations. Just see, there is just a bunch of conformations which the aspartate can adopt and a bunch of conformations which phenylalanine can adopt. Okay, whereas in DFG active, DFG inactive, just see phenylalanine, this plot, those who are aware of organic chemistry, this plot is phi-psi plot and this represents the dihedrals of the backbone. Okay, so look at this. In an inactive conformation, the phenylalanine can have so many different phi-psi angles, but in an active conformation, it has only a bunch. So that proves the statement the inactive kinases can be much different, whereas the active kinases can be only similar to each other. So what makes inactive kinases to adopt more conformations? Okay, what happens in an active kinase? The aspartate is towards the ATP binding site. Okay, why it is towards the ATP binding site? Because it can stabilize the ATP kinase complex through magnesium ion coordination. Okay, because of aspartate, the magnesium remains in position and then the phosphorylation can take place. There is no such restriction for the inactive kinase. Okay, so the inactive kinase can adopt several distinct conformations by changing the dihedrals. Okay. Coming to type 2 inhibitors, just now I mentioned that can be in several distinct conformations. Okay, and they type 2 inhibitors bind to kinase in a DFG out conformation, C helix out conformation. Okay, these compounds not only occupy ATP binding pocket but extend to the adjacent allosteric pocket. Let us see this. I'll first explain this and then go to these two slides. See, this is the ATP binding pocket. Okay, this is the allosteric pocket. Okay, and this is the DFG motif. Now look at the type 2 inhibitors and how they are binding. This is ponatinib, ATP binding pocket, the DFG, okay, and the allosteric pocket. Okay, now remember they are still binding to ATP binding pocket, and so they are still ATP competitive. Look at nilotinib. Okay, look at these regorafenib, sorafenib. Everywhere there is a ATP binding pocket to which part of the compound binds. There is a DFG motif to which part of the compound binds, and an allosteric pocket. This allosteric pocket is accessible only because C helix has moved. Okay. Now, a very nice design strategy which was applied using computer aided design as well as the other parts of design. See, this is imatinib, the first type 2 inhibitor of BCR-ABL kinase which came into practice, clinical practice. When computer aided drug design was invented and when the crystal structure was also known, it was realized that imatinib, can you see these dotted lines? And you will realize that imatinib doesn't fit exactly into the pocket. Okay. Now, since imatinib doesn't fit exactly into the pocket, scientists thought of changing the imatinib structure. Okay, an extensive SAR was carried out, structure activity relationship, and this resulted in nilotinib which fitted in the pocket perfectly. Can you see this picture where nilotinib fits into the pocket completely? Now, how did that happen? Using the crystal structure, using the computer aided drug design, scientists realized that this area of imatinib had a wrong conformation. It could not fit into this pocket, okay, this pocket, and so they tried to change it and they realized that imidazole fitted well into that pocket. Additionally, have a look at this once again. There was an area left over here. This was a small hydrophobic area. And to address this small hydrophobic area, they attached a CF3. Now, when they attached a CF3, they checked it first through computer-aided design, and then they synthesized this compound, nilotinib, and they found that nilotinib has better activity than imatinib. Okay, so now things are fitting, they are falling in place. Okay, so far, so good. Let us try to understand what are type 3 inhibitors. As I mentioned earlier, these are the inhibitors which bind to an allosteric site, but this allosteric site is near the ATP binding site. Okay, they do not occupy the ATP binding site, but they occupy a site or an area near the ATP binding site. Okay, and because they do not occupy the ATP binding site, they exhibit high degree of selectivity. I hope everybody understands that the receptor, the kinase receptor, is so much similar in its active site. Every kinase is similar in its active site, but the allosteric site differs in every kinase. And because the allosteric site differs, the allosteric or the type 3 inhibitor will be able to bind to the target kinase selectively. Okay, and this binding cancels or sort of changes the agonist function. Now the ATP cannot bind, and therefore phosphorylation of protein does not occur. Look at this figure, you will realize that this is the ATP binding site. Okay, this is the N lobe, which is mainly consisting of beta sheets. This is the C lobe. Apart from loops, there are alpha helices, and the yellow one is the ATP binding site. Okay, the pink one is the allosteric site. Okay, and inhibitor which is shown over here is cobimetinib. There are other inhibitors which are in clinical trials, and one is approved, that is trametinib. All these inhibitors bind to the allosteric site, and so they will not be ATP competitive. Okay, increasing the concentration of ATP is not going to reverse the action. Here are structures of type 3 inhibitors shown. This is cobimetinib, and you must have realized that there is no nitrogen containing aromatic ring in these structures. Why? Is because these structures do not bind to the ATP binding site. The ATP binding site is very well designed to accept a nitrogen containing aromatic heterocycle like purine. And so these compounds are structurally different. Okay, now we come to type 4 inhibitors. As I mentioned earlier, type 4 inhibitors bind at a site far away from the ATP binding site. Okay, I have shown here in this figure nilotinib. Nilotinib, if you recollect, is a type 2 inhibitor. Because it is a type 2 inhibitor, it will bind to ATP binding site, and it will also bind to the back pocket. Okay, this is the binding ATP binding site, and look at asciminib. Asciminib binds so much far away from the ATP binding site. Okay. The structure is also shown over here, asciminib. Okay. Now, what does asciminib do? Asciminib binds to a site which is myristoyl binding site in the C lobe. Okay. Myristoyl binding site in the C lobe. Some of you who have who recollect biochemistry may recollect that myristylation is a very important post-translational modification. And for that, there is a myristoyl binding site to which myristoyl residues bind. Okay. Now, this myristoyl residue is present in wild type ABL and it binds over here. Okay. What happens in wild type? I hope everybody remembers what is wild type enzyme. Wild type meaning non-mutant enzyme. Okay. So if there is non-mutant ABL, then there are myristoyl residues which bind here. Okay. And this binding of myristoyl residues causes bending of this helix, which is shown in green, to over here. Okay. This helix bends and comes over here. Now, when the helix bends, I mentioned earlier that in BCR-ABL, there is continuous activation without a ligand. Also, it is permanently activated receptor, and therefore it causes permanent cell division to occur without any control. Okay. Now, that control is achieved by myristylation, and this myristoyl residue is absent in the mutant BCR-ABL, and so the myristylation does not occur. Because the myristylation does not occur, the helix cannot bend. Okay. Now, what does this compound do? Asciminib. Okay. Asciminib binds at this area. Okay. Asciminib binds at myristoyl binding site. Okay. And therefore, it cancels the auto activation of BCR-ABL mutant protein. Okay. BCR-ABL mutant protein is auto activated and therefore brings about lot of cell division. Now, this asciminib binds over here. Because asciminib binds over here, the auto inhibition is imparted, or auto activation is lost, and this is beneficial in chronic myelogenous leukemia. Imatinib functions as a type 2 inhibitor, whereas asciminib here functions as a type 4 inhibitor. Okay, so if there is any resistance and imatinib cannot bind, asciminib still functions. Okay. Now this asciminib again was designed by fragment-based design coupled with computer-aided design and NMR monitoring of drug receptor binding. Okay. These were two fragments. Fragment-based design, what is done is some fragments which have possible, although low activity, are taken up and they are systematically built further. New moieties are added, they are built further and then checked. Okay, so these two fragments were identified. They were binding to the enzyme. They were binding to the kinase, but not very strongly. So, scientists wanted to change these. Now look at these two fragments. They altered it, retaining the two halogens over here. They took the NH from here, CO from here, and built a higher molecule. Okay. This higher molecule was built, but it was still inactive. Further change the higher molecule. Now look at the next molecule. This entire part is same. Instead of two halogens, what they have done is inserted one ring, chain over here, CF3O. What happened? The molecule suddenly became active, moderately active. They wanted to change it further. Okay. In the next thing, what they did, computer-aided design, they realized that there is a lot of steric hindrance because of this methyl, and so they changed it to morpholine. Not only that, these two were removed. Okay. And look at the GI50, which improved from 8 micromoles to 2 micromoles. Still not satisfactory because it has to be in nanomoles. Okay. So further modification, this part was retained. Now, what was remaining was only this, correct? This ring had to be modified. So this they modified. They must have synthesized hundreds of analogs, and finally this worked. Instead of benzene, they put a pyridine ring and a alicyclic ring over here. That was retained, but now here the pyridine ring which was there was changed. Okay, this entire part was changed, and this resulted in that clinical candidate assimilate. So slowly they were going on building, altering the molecule till success was achieved. This became a candidate. Okay, now coming to type 5 inhibitors. All protein kinases are bisubstrate enzymes. Okay, how? There are binding sites for ATP and there are binding sites for the substrate which needs to be phosphorylated. Okay. And also remember, the cell inside it has about 1 lakh of proteins, but the kinase selects the specific substrate for phosphorylation. Okay, the kinase does not go about, and tyrosine, threonine, serine are present in all these proteins, but kinase selects a specific protein for phosphorylation. Okay, this implies that there is a specificity of the substrates. Okay, kinase is very specific about the substrate which it phosphorylates, although the ATP binding site is same. So the next thought process was, why not target that substrate binding site? What they did, one part is ATP binding pocket, the other part is either the substrate binding site or any other nearby site. So they designed inhibitors where one part of it can bind to the ATP binding site, the other part can bind to a faraway substrate binding site, and these two molecules were joined by a linker. Okay, one molecule substrate binding site, other secondary binding site, they were joined by a linker. Okay, linker is a chain, sort of chain, which you can decide its length. It can be CH2, CH2, CH2 or CONH, CH2, CH2, CONH or any such thing. Okay. Sometimes the linker joins the ATP binding ligand to a pseudopeptide, and this pseudopeptide can bind at the substrate binding site. Okay, instead of directly attaching to some linker to the substrate binding site. An example over here. This is an ATP analog. Can you see this is a purine? This is a sugar. There is no phosphate, but this can bind to ATP binding site. Then look at the linker. There is NH, CH2, CO, there is lysine NH, again along, and at that other end, it is attached to a peptide. Okay, this peptide is not the original substrate at the substrate binding site, but it can bind to the substrate binding site and can lock the substrate binding site so that original peptide doesn't bind. A typical theory of inhibitors. Okay. So, it can achieve a very high potency and high selectivity. Why? Because using this substrate binding site, you are modulating that one particular kinase only. The last type of inhibitors are covalent inhibitors. Okay, people are usually scared because alkylating agents in cancer have a lot of side effects. But there are other common covalent inhibitors which are used. Penicillin is a covalent inhibitor, aspirin, omeprazole, okay, cephalosporin. All these are covalent inhibitors, and they have not caused as much problems as alkylating agents cause. Okay, but covalent inhibitors were not discovered by directed efforts. Okay, most of them were discovered by serendipity or by chance. Okay, covalent inhibitors usually people associate with toxicities, that is hepatotoxicity and idiosyncratic toxicity. But covalent kinase inhibitors have worked. Just try to understand what are covalent kinase inhibitors. They form a covalent bond with kinase. Okay. Now, for any compound to form a covalent bond, it must have an electrophilic group. Okay. And this electrophilic group can react with some nucleophilic site on the protein. The easiest nucleophilic site on the protein is a cysteine, which is present. S has two lone pairs of electrons, and so it can act as a very good nucleophilic site in the protein kinase field. So, if a cysteine is a non-conserved cysteine, non-conserved meaning not common to multiple kinases. If there is a cysteine and that cysteine is not common to multiple kinases, then if that cysteine is targeted for covalent inhibition, then selectivity will also be there. Okay. So, depending upon which cysteine is targeted, they can increase the selectivity. Covalent inhibitors can increase the selectivity even if their intrinsic reactivity is low. Okay. This is just information. See, the covalent inhibitor related patent applications, with just see how much sharp increase there has been from 2015 to 2020. Likewise, covalent inhibitor related papers, there has been a rise, and that rise has been since maybe 2012 onwards. Okay, and that is quite logical. If people have started working from 2012 onwards, and then finally the patent granted can be from 2015. Here is an example. This is one investigational drug. Okay, this drug has an electrophilic group. Okay, this electrophilic group reacts with cysteine. Now what happens? KRAS is a kinase kinase enzyme. Okay, kinase protein. Okay, this electrophilic group can form a bond with mutant KRAS which has cysteine. The original KRAS or the wild type KRAS doesn't have cysteine, but the mutant has cysteine. So it has opened up a very good opportunity for covalent inhibitors, because this covalent inhibitor is going to react only with the mutant. And so that means only with the cancer cells. Okay. So this offers selectivity to a very, very great extent. Just see some common covalent scaffolds. The alpha beta unsaturated ketone is the most commonly used. It can be used in combination with amide or only ketone. Okay, there are cyanoacrylamides, there are fumarates, everywhere it is alpha beta unsaturated ketone. Okay, but sometimes like this epoxides or alkynes which can alkylate any protein very well are also used. Okay, nitriles, alkynes, these are the triazoles, 1, 2, 3 triazoles. Regarding covalent inhibitors, the older design strategy was attaching an electrophile to non-covalently binding drugs. I'll show in the next example. This is ibrutinib. This has to bind to the ATP binding pocket. Okay, and there was a additional electrophilic component which was attached to it. Similarly, afatinib, this is a typical structure of ATP binding component to which an electrophilic component has been attached. But since then things change, and now even attaching other parts of the molecule to the electrophilic scaffold is also done. Okay, for example, in this case, the other parts of the molecule were attached to the electrophilic scaffold. Okay, what is shown over here is modifications of the existing drug. These are done and the triple bond is introduced. Just have a look at it. This is ibrutinib and this is a analog of ibrutinib with an additional triple bond. Now this additional triple bond serves to determine, it is a very good alkylating agent and therefore serves to determine how many proteins in a cell are alkylated. Okay, so it serves to determine what are going to be off-target alkylations, the undesired alkylations. Okay, so I think we have come a long way starting with imatinib. First were the type 1 inhibitors, but majority of the problems because of non-specificity, non-selectivity. Then came the type 2 inhibitors, which target an additional pocket, an additional binding pocket. Okay, but still ATP binding was maintained. Then came type 3 inhibitors, where there is a completely different binding pocket. Okay. Then came type 4 inhibitors, which were bound to a remote binding pocket, and the mechanism was completely different. Then there were type 5 inhibitors, which were binding to two different pockets. the binding to the second pocket led to a different mechanism than binding to ATP competitive pocket. And then there were covalent inhibitors. But in all this process, all the while, it has been realized that I mentioned all the active sites of kinases are similar, so that they can bind each other's inhibitors. In other words, the kinases are promiscuous. They do not stick to one inhibitor. The other kinase inhibitor can also inhibit the first kinase. Okay, so let us try to understand why the kinases are promiscuous. And it has been realized that some kinases are more promiscuous than others. So type 1 kinases, we said, because of lot of structural similarity in the active site, and there are salt bridges which maintain the active conformation. And so the active conformation is so much similar that any kinase inhibitor of type 1 can bind to any of the kinase. In type 2, the structures are different, the residues are different, but there are some evolutionary conserved residues. Evolutionary conserved residues meaning from the non-vertebrates to lower vertebrates to mankind. Till here, the residues are conserved. Okay, so since there are conserved residues, these conserved residues contribute to promiscuity of kinase. They help the other inhibitor to bind to the first kinase. 8 kinases have been identified as particularly promiscuous. Okay, and these are mentioned over here. Out of these, whatever I have written in green, meaning these are involved in cancer to a greater extent, these are not much involved. Okay, so these are thought to be promiscuous kinases. Now what happens because of promiscuity? Inhibition of multiple kinases leads to off-target side effects, but inhibition of correct combination of kinases may be a very effective treatment of cancer. Okay, if there are 3 particular kinases which are going off target, okay, which are firing continuously, which are activated, and you are able to inhibit only these 3 kinases, then it can be very effective treatment of cancer. Let us try at the end to understand what are the current challenges in kinase inhibition, discovery of kinase inhibitors. First is the lack of understanding of disease process. Cancer, although there are a lot of studies which are done, cancer is still difficult to understand. Which combination of kinases goes haywire has not been ascertained in all types of kinases, and therefore sometimes we don't know what is the target kinase. Okay, then there are limitations on the computation. A lot of computer aided drug design work has been done in kinases, but there are many limitations on the computation. One very simple thing I will tell you is when we started working on CDK2, we realized, we thought we will take a crystal structure, we will dock our compounds, the library into that, and we will go ahead. When we looked at the crystal structures, there were 350 crystal structures. Now, which one to take? That was always, and this arises in case of many kinases because inhibitors belonging to different chemical classes give different conformations of kinases, and so it is difficult to decide which one to start. Then the entire DFG flipping and the C helix movement people have been able to model it, but not very effectively. And this causes a lot of limitation. Apart from the ATP binding site, the other pockets which are there, they are quite shallow. And shallow pockets are not very amenable to inhibitor development. Okay, so that is a limitation of type 3, development of type 3, type 4 and type 5 inhibitors. Most of the pockets are shallow pockets. Okay, and the chemical space of inhibitors is getting exhausted. A lot of chemical space has been tried already. So not much new in terms of chemical space can be done now. So these are all the current challenges. Coming to future prospects, new websites and new softwares have been developed which are able to predict which chemical scaffolds will be more effective generally as kinase inhibitors, not very specifically in that, but generally. So that is one of the very simple advancement. Improving selectivity and reducing off-target effects. It has been attempted via a variety of ways. One of the ways is exploring alternate allosteric site. Disease process has been explored to a very great extent and is being explored. Computational aspects are improving day by day, and all this. Apart from that, people are now talking about combination of kinase inhibitors with immunotherapy. I think all of you know in 2018, the Nobel Prize was won for immunotherapy for cancer. Okay, so the people are now talking of combining this. In the end, I will like to conclude that we are only at the beginning of the era of targeting protein kinases for therapeutic purposes. Although about more than 100 kinase inhibitors have come, we are still at the beginning. We are not aware of everything that happens in a kinase, and we have not explored kinase. So although they have come a long way from imatinib till the covalent inhibitors, they have expanded in multiple directions, but future will see many more and more effective drugs to fight the dreaded disease that is cancer. And doesn't stop at that. People are investigating kinases for multiple diseases, including diabetes, including inflammatory conditions, and even as antimicrobials. I think all of you know that efficacy of antibiotics is being questioned now in next 20 years, and therefore people are exploring kinase inhibitors. So with this, I thank everyone for patiently listening to a talk which was at a different when people must have been exhausted since morning with a very creative and good talks, but I hope I have tried my best for this. With this, I thank you all and open for questions if any.
Thank you, ma'am, for a wonderful presentation. We will take questions now, and then I will sum up the session. So any questions? I have one in the chat box from our own faculty, Dr. Archana Gurjar. She commends you on this wonderful presentation, and her query is, in fragment design, slide number 46, lot of halogen substituents, fluoro, chloro are present. Can you comment on toxicity profile of these compounds?
Yes, the toxicity profile, chloro and fluoro are present, but chloro and fluoro in kinases have been used because they can fit in the lipophilic pocket first, and secondly, the kinase inhibition causes, I mean, usually they weigh the risk benefit ratio. So when you compare the risk benefit ratio, yes, the kinase inhibitors are more beneficial, I will say.
Well, I'm not seeing any more questions in the chat box. Right? Please do put in your questions, and otherwise, if anybody wants to ask on the mic, they're free to do so. Please do it. Any questions? Any further questions? In the meantime, ma'am, I have a couple of questions.
Huh.
As you said that at the moment they are used for cancer therapy, and many more applications are in the offing.
Yes.
They can be used for anti-inflammatory conditions and for immune therapy, immune diseases. What about some neurodegenerative diseases, ma'am? Alzheimer's or even Parkinson's? I believe our kinases are, I think they have a role in liberating interleukins. So inhibiting that aspect would help, ma'am?
But I have not come across in my reading about the neurodegenerative. Multiple papers I came across antimicrobials, anti-inflammatory, diabetes. There are about 3 or 4 kinases, 6 kinases, I suppose, which are identified. But I have not come across, or probably I did not look into neurodegenerative diseases.
Ma'am, it may not be there. I'm just asking aloud. I'm just expressing my thoughts. Is it possible?
Yes. It is. I have not come across.
Okay. So at the moment, the therapy exists only for cancer. So all these inflammatory, for inflammatory diseases, for immune disease, it's still in the pipeline.
Yes, and monoclonal antibodies which bind to the ligand binding region of so many growth factors.
Yes, ma'am.
Finally, it is not kinase inhibition, yes, but the growth factors, the monoclonal antibodies.
Yes.
And the immune response, there are monoclonal antibodies.
Yes, ma'am. One more question from my side. Can we see resistance to the protein kinase inhibitors in future? I mean, are they really curative, or they're only delaying the progression?
Resistance has been reported. Imatinib was the first drug, and imatinib resistance has been reported, and that is why they shifted to nilotinib, where designing strategies were applied and all that. But resistance is seen. EGFR inhibitors also resistance is seen. VEGFR inhibitors resistance is seen. I don't know whether we can call it only delaying things, because cancer cells themselves are so aggressive. Under pressure of the drug, mutations can occur, and these mutations usually result in loss of drug binding, plus some PGP is also active in case of cancer cells, which throws the drug out. So we can say that this is the first step probably.
Right, ma'am. But will they be curatives, or they're only delaying the progression of cancer?
No, in fact, after imatinib and after nilotinib now, in case of some patients, the life expectancy was reported to be equal to a normal individual. For CML, that is chronic myelogenous leukemia.
Yes.
Whereas mutations are seen only in some patients and the reasons for these mutations are not known. Every patient doesn't show the mutations.
True, ma'am. Okay, any further questions? By Vaibhavi Garge, I think I was seeing your hand up. Right? Okay, Akanksha, may I request you to hand over the mic to her?
Allowed mic to her.
Yes. Vaibhavi, please go ahead with your question. Okay, in the meantime, there's another question from Dr. Vrish. His question is, how target delivery of anti-cancer drugs can be achieved?
Yes, target delivery of anti-cancer drugs is there are a lot of receptors which have been tried so that the anti-cancer drug is delivered only to its target. But there are a lot of teething troubles in that also. One way of targeted delivery is the delivery, like I explained, the covalent inhibitors targeting only the mutant cystine of cancer cells. So they enter other cells, the normal cells, it is not going to be affected. So in that way, this is a chemically, it is not targeted in the true sense of it because other cells will uptake it, but the effect may not be very prominently seen. There are others, like there are some mannose receptors, there are other receptors which have been considered in delivery of these drugs, and all these kinase inhibitors, most of them belong to BCS class 2. So even otherwise, formulation considerations will be there. There is one more question. The mechanism of resistance for kinase inhibitors common? No, mechanism of resistance can vary. Essentially, two different mechanisms of resistance. One is P-glycoprotein throwing away the drug from the cell or not allowing it to enter the cell. That causes resistance. The second reason of resistance is the drug not able to bind to the receptor because of mutation. But a third way of resistance is the cancer cells developing an alternative pathway. And so the mechanism of resistance for all kinase inhibitors will be different.
Are there any further questions? Should we wind up the session, ma'am?
Yes, we can. If there are no more questions, we can wind up the session. And thank you so much, all the participants, for patiently listening at this time of the day. Thank you so much.
Thank you, Urmila ma'am. It was indeed a very nice and informative session. Thank you, Vandana Panda ma'am, for chairing the session so nicely and maintaining the enthusiasm. I thanks all the participants for being with us for the entire day. We will see you tomorrow at same time, 9 a.m. And the session has been shared with all of you. Please give the feedback for today's session. Thank you, all of you. Thank you for being
Akanksha,
with us. Yeah.
I still have to sum ma'am's presentation. We do it at the end of the questions. Right. Okay. Thank you, ma'am, for a lucid walk through the world of protein kinases and their inhibitors. Ma'am, you have explicitly expounded the different type of kinase inhibitors, the development of this cutting edge class of compounds since the introduction of Imatinib, the first tyrosine kinase inhibitor used to treat a number of human leukemias and tumors. Small molecule kinase inhibitors are indeed an exciting field of research set to stomp chemotherapy. You have successfully instilled in us and understanding about kinase inhibitors, different conformational states of the kinases, types, and the approaches adopted in drug discovery to make them more selective and effective. Thank you, ma'am, for a wonderful presentation. With this presentation, we end the scientific affairs of day 2 with a promise that equally exciting topics await you tomorrow. Thank you so much.
Thank you,
Thank you,
Dr.
Vandana.
Vandana.
Thank you all once again.
Thank you, madam. I have to do one important announcement for all the attendees. Some of the attendees are writing to me that they have missed the morning attendance. So I will be posting the morning attendance link again back into the WhatsApp group, which will last only for 15 minutes. Those who have missed their attendance, please fill it immediately. Thank you.
Thank you, Mrinal. Thank you, all of you.